In this video, I will show you some tips of using the tables in the section of differentially expressed genes. And in this section, you can check the heat map, Volcano plot, and the differentially expressed genes, and the alternative splicing, the MA plot, PCA plot, and co-expression analysis by WGCNA software. In those two tables, I also embedded the functional annotation from UniProt keyword, UniProt pathway, RNAseq summary that from the UniProt database, and the KGG, GO, and the GeneRef. The GeneRef is from NIH database, and also provided the homology genes of human. The human homology genes, and the DEGs2 module estimated gene expression level and the standard error. The gene type distribution could be checked from here, the gene bio tab. If you want to see all long non-coding RNA, you could just filter gene bio tab from here, and then you will get finally 85 significant changed long non-coding RNA. And you can further use the filter here to get how many long non-coding RNA is upregulated, and clear for filters here. And for using the filters, you can learn more from here. Then if a gene has significant alternative splicing event, for example here, you can click the link and to check the details of the significant changed alternative splicing. If the alternative splicing also with the alternative if the alternative splicing also with the differentially expressed exon, you will see the differential exon usage test results. And for more details, please check their original software, which is isoform and switch analyzer and DEGs, to understand all column from the results. Here I will show you an example to use the functional annotation embedded in this table. For example, if we want to check whether or not there's a transcription factor that related to calcium signal pathway, we can do this search tabs to filter out our target genes. Let's check the GeneRef and UniProt keyword. And in the UniProt keyword, we can use these three keywords to define a transcription factor. Transcription regulation and DNA binding. Then we got all significant changed transcription factor in this list. There are totally 65. And then we can search the calcium related gene from the gene reef. Now we get 4 transcription factor that are related to the calcium things. And then we in the gene reef column, the number here is PubMed ID, the article ID in the PubMed database. So the calcium should be highlighted in this column, for example here, and then we can copy this PubMed ID to check the original paper. And the sentence follow the PubMed ID is the manually summary of that paper that's related to this gene. Then in the co-expression analysis, you can find the genes that shows co-expression pattern. So this is actually a cluster analysis. For example, if we for example, this cluster means all those genes are positive related to your treatment difference and significant. And we can check these genes from this table. And if you want to check what functions is enriched in those genes, you can triple click them and copy it to Excel. And split them using the text to columns function in the Excel software. Then you can copy these genes into David's functional enrichment website tool. To check the functional enrichment result. Like this.